Ever try to build something really cool out of Legos? Like really complicated?
Yeah, definitely. Back in the day, I used to love those huge Lego sets.
Right. And you're thinking, man, if only I could do this with like DNA.
Haha, okay. Maybe not quite the same.
Okay, well, maybe you haven't thought that, but that's kind of the basic idea behind this thing called Golden Gate Assembly. That's what we're diving into today. And get this, we've got this awesome blog post from TeselaGen Biotechnology. Apparently, they are doing some really interesting things in this space. So yeah, we're going to unpack all that, figure out what Golden Gate Assembly is, why people even care, and
And those 4 things to look out for. I read that blog post. It's a good one.
yeah, those 4 things. The blog post had some pretty strong opinions about those. And it seems like something anyone who's actually, doing this stuff in the lab would definitely want to know.
For sure, this isn't just theoretical stuff. This is like real-world application kind of advice.
Exactly. So let's start with the basics. For anyone who maybe hasn't gone full-on mad scientist in the lab lately, what exactly is Golden Gate Assembly?
So picture this.
Yeah.
It's like you've got these molecular scissors, right? They're called restriction enzymes, and they can cut DNA at very specific spots.
Okay, so we're talking about chopping up DNA like it's, well, not like it's Legos, but more like...
Sure, like a very precise, very tiny pair of scissors.
Yeah, exactly.
Okay, so we've made our cuts, and now we've got these little sticky ends on the DNA fragments, kind of like puzzle pieces. That's where ligase comes in, another enzyme. It's like the molecular glue that binds those sticky ends together, and boom, you've assembled your DNA fragments in the exact order you wanted.
Okay, I'm starting to see how the whole assembly thing works here, but like, what are we actually building? What's the point of all this cutting and pasting DNA?
That's the really cool part. We're talking about engineering organisms to do some pretty incredible things, like imagine organisms that can produce life-saving medicines.
No way. So we're talking like new drugs and therapies, all thanks to this Golden Gate Assembly thing.
Exactly, and that's just the tip of the iceberg. We could create entirely new biomaterials. Imagine like super strong but completely biodegradable plastics.
Okay, that would be a game changer.
Right. And even in agriculture, engineering crops that can thrive in tougher conditions, think droughts or floods to help secure our food supply.
Wow, this is way more than just some cool science experiment. We're talking about things that could seriously change the world.
Absolutely.
But TeselaGen's blog post isn't all just, hey, look at this cool new tool. They get into some pretty specific challenges, right? Like, they're not just saying it's easy.
Yeah, they don't sugarcoat it. They lay out these 4 things to look out for, which honestly, from my experience, are absolutely crucial for anyone who's serious about using this technique effectively.
Okay, so let's hear it. What's the first thing we got to watch out for? Lay it on us.
All right, so imagine you're building a house, right? You wouldn't just start laying bricks without a solid blueprint, would you?
Definitely not. I've seen enough home renovation shows to know that's a recipe for disaster.
Exactly. And the same principle applies here. Meticulous design is absolutely essential for Golden Gate Assembly. You need to map out exactly where you want each DNA fragment to go and make sure those restriction enzyme cut sites are perfectly positioned.
So one wrong cut, and the whole thing's toast. That's a lot of pressure.
Yeah, precision is key, but that's where the second thing comes in. Choosing the right enzymes for the job. It's just like any other tool, really. You wouldn't use a hammer to tighten a screw, right? You need the right restriction enzyme for each specific DNA sequence.
So it's all about using the right tool for the right job. But even with the perfect blueprint and all the right tools, sometimes things still go sideways.
100%. And that's where the third thing comes in. Being prepared for errors. Even with meticulous planning and the best enzymes, you might get mutations, or fragments might join in the wrong order. It's kind of like when you think you're nailing those Lego instructions, but suddenly there's a piece left over and you're thinking, where did this come from?
Am I losing my mind? Was this piece always here?
Right. And in this case, that stray Lego brick could be a pretty big deal. That's why TeselaGen really emphasizes the importance of quality control. You've got to be checking your work as you go, making sure everything's lining up the way it should.
So constant vigilance. Don't just assume everything's going perfectly.
You got to catch those errors early before they snowball into something bigger.
Okay, so we've got our blueprint, we've got the right tools, we're checking for errors like hawks. What's the final thing to keep in mind?
It's all about troubleshooting. Even with all the precautions, sometimes you just get unexpected results.
Yeah.
It's science.
That's the name of the game. Yeah.
Exactly. And that's when experience and a deep understanding of the process really comes into play. You got to know how to interpret those unexpected results, figure out what's causing the issue, and come up with creative solutions. It's all part of the process.
So Golden Gate Assembly, a powerful tool. Huge potential, but definitely not something you can just set and forget.
That's a great way to put it.
You need a solid plan, the right tools, a watchful eye, and a good dose of that problem-solving spirit. But hey, if you're ready to roll up your sleeves and dive into the world of molecular Legos, then by all means, go for it.
Yeah, and who knows, maybe you'll be the one to discover the next big breakthrough in medicine, materials, or even agriculture.
There you go. That's the dream right there. This has been a fascinating deep dive into Golden Gate Assembly, and frankly, it's pretty inspiring to think about all the possibilities this technology unlocks.
I agree. It's a really exciting time to be working in this field.
Thanks for joining us for this deep dive.